All right, so we covered some of this in class the week before Thanksgiving. I'm going to hit it kind of quickly, but this is the last PowerPoint lecture that's going to be recorded, obviously, for the semester. This is the part, let me scoot this up, that we covered in class, in addition to a couple other slides. I'm not going to go into a lot of details on this. If you weren't in class for this, then it's just like, you know, any other absence that you would have missed. So we talked about that solutions are mixtures of two or more substances where the molecules of those substances are evenly distributed. We're going to deal with water being the universal solvent, of course, in this case, and talk about tons of different situations where organisms are in different types of solutions, or cells are in different types of solutions based on the environments in which organisms live. So water is going to be the universal solvent. It's polar, and it's going to dissolve any type of solute that is also polar or ionic. Solutions are going to have different concentrations depending on how much solute is dissolved in the solvent. We're going to deal with this mathematically to some extent, but also just kind of talking in general, that is the solution concentrated or dilute in comparison to another solution. And then we talked about that specific example of frozen versus liquid orange juice in class. Okay, so we already defined this definition, concentration is the mass of solute in a given volume of solution. So you've got an example there that if we dissolve 12 grams of salt in 3 liters of water, then we get a concentration of 4 grams per liter. In class, we talked about these two different solutions as well, 20 grams of salt in 4 liters of water, or 30 grams of salt in 5 liters of water. And that the second one, the 30 grams of salt in 5 liters of water, would be more concentrated than the 20 grams in 4 liters of water. So again, if you weren't in class, we just did the math on this. I would suggest that you do so, okay? And then write that down, and then you'll see, hopefully, that this is the more concentrated solution. A concentration gradient, by definition, is just the difference in concentration between two solutions. Again, we will deal with this to some extent mathematically, but we will also just deal with this kind of relatively, saying this solution is more concentrated, like solution A is more concentrated than solution B. We also already discussed this, that the plasma membrane or cell membrane is also called the phospholipid bilayer. It's semi-permeable or selectively permeable, meaning that it only allows certain substances to move in and out of the cell. It surrounds and protects the cell. It's made of 2 layers of phospholipids with proteins that are embedded in them. And as you were supposed to have addressed in your fluid mosaic model worksheet and watching the short video clip on Edpuzzle, that the membrane is flexible. It's fluid, which means that it's, you know, constantly changing shape and moving. It is going to be composed of phospholipids, proteins, and carbohydrate chains. We also already covered this slide in class. A phospholipid kind of looks like this little balloon with 2 strings. It's a molecule consisting of a phosphate group, which this is the phosphate, which is PO4, and then 2 fatty acid chains. The phosphate group is polar, and it's hydrophilic. It likes water. The fatty acid chains, those 2 tails, they are non-polar, and they are hydrophobic, and they are afraid of water. So these get organized, and there's another picture on the slide beneath this. These get organized in such a way that the non-polar fatty acid chains that make up the tails, that they face each other inside the middle of the phospholipid bilayer. The heads are on the outside, and that's because they are polar, and they are going to be attracted to water, which is also polar. So the non-polar tails, that is what specifically separates the cell from its surroundings. This is what these non-polar tails, this is what dictates what can move across the membrane and what cannot move across the membrane. Okay, it's truly kind of the barrier. The heads are not. Then there are proteins in the membrane, which on the fluid mosaic model worksheet, you saw that there are different types of proteins. They form channels or pumps and receptors, and they move materials across the membrane. We'll deal with that in a little bit more detail with a couple of specific things with this. We're going to focus on some other things, though, as far as just how materials move across the membrane in more detail. Carbohydrate chains, they are chemical identification cards, and they let cells identify each other. So in class, we talked about this particular picture. This is basically just a snapshot in time. That kind of, if you imagine that if you blink, okay, that in just a minute, that let's say, for example, that this protein with those carbohydrate chains on it, that maybe it's going to go way back to this back region back here. If we kind of imagine this picture being three-dimensional and the cell going kind of backwards in space, as opposed to this being a flat picture. And then maybe a minute later, this protein shifted over. Maybe this one shifted up to here. Okay, that again, the membrane is constantly changing, and it's never the same, okay, because it's a living organism, it's dynamic, it's changing, it's a mosaic of these different substances, the phospholipids, the proteins, and the carbohydrate chains. And the proteins are polar, I forgot to say that, and it's listed there next to that structure. All right, so this next slide I'll go a little bit slower on. I did that other stuff that we'd already covered in class a little fast because of just that we had covered it in class, but I figured I'd just put it all into one lecture. So transport mechanisms, these are going to take place so that cells maintain homeostasis. So as I mentioned in class before Thanksgiving, this is the unit where we are going to focus on homeostasis. There's two major categories of transport: passive transport and active transport. So by definition, passive transport is the movement of a substance through the cell's membrane without the expenditure or the use of energy. Okay, so substances are going to move with the concentration gradient, down the concentration gradient, or along the concentration gradient. We can kind of put those two words down and along where it says with. Okay, on the flip side, active transport, this is movement of a substance through the cell's membrane with the expenditure of energy. So the substance is going to the movement of the substance is going to require the cell to actually use some energy in the form of ATP. So the substance is going to move against the concentration gradient. Okay, so again, because of the fact that this is requiring energy, because of the fact that this is moving the substance against the concentration gradient, the cell is going to use energy. Okay, so the reason for this is because the substance, whatever it happens to be, is going to move through the membrane in the opposite direction from what particles are typically going to do, and we'll talk about what that means in just a second. Okay, I like to give the analogies, or a couple of different analogies for this. So passive transport is going to move the substance from high concentration to low concentration. Okay, so it doesn't matter what the substance is, it's going to go from high to low. Oops, sorry, I wrote the wrong word. High to low. Okay, for active, so I would suggest that you write some of this stuff down. Active transport is going to move the substance from low to high concentration. Okay, and we'll just put in concentration here and here as well. Right, so as a result, one way to think about this is that if you are a kid and you go to the playground, okay, then you're going to expend a lot of energy to walk up the stairs that then get you to a slide, to slide down the slide. Okay, so it takes more energy to climb the stairs, okay, going from low on the ground to high, right, to get ready to slide down. Going down the slide is like passive transport. Okay, you start at a high point, you go down toward a low point. This takes you way less energy. You just kind of have a little push and you get down the slide. Okay, or this is another example I always give with this, that I live at the bottom of a really steep hill, so I'm going to expend a lot more energy to go on a walk and walk up the hill from a low point to a high point. Okay, it's going to be easier for me to walk down the hill when I return from my walk from the high point to the low point. All right, for this slide, I actually want to start with this bullet point that says particles are constantly in random motion. So hopefully you learned that in middle school. In general, that means that a particle is more likely to move from where there are many of those particles, whatever they happen to be, to where there are only a few, if any, of them. So when there's a lot of particles, like here on the left on this diagram, they tend to move to where there are few of them on the right, okay? So that being said, passive transport is going to move some sort of substance or particle from high concentration to low concentration. There's several different types of passive transport. The first of which is diffusion. Okay, so by definition, this is the net movement of particles from an area of high concentration to an area of low concentration. Net means kind of an overall flow, okay? And I'll mention what that means in just a minute compared to just not using the word net, right? So that being said, hang on, let me go ahead and just switch this to this next picture and slide. Okay, so on your slide, these are not red and blue dots, okay? But we've got a solute in red here and a solvent in blue. So obviously our solvent, okay, is going to be water. Okay, it doesn't really matter what our solute is, okay? In general, particles are going to tend to move from where there are many of them to where there are very few of them, right? And so by diffusion, if this is a beaker of water, for example, right, and we drop a red dye into that, this is a classic example to look at the movement of particles by diffusion. We drop that red dye over here on the left of this beaker. There's no red dye particles over here. Slowly, these dye particles are going to move across that beaker, as we can see here, until they fill that space uniformly, right? This is going to, so diffusion is going to occur until equilibrium is reached. I want you to add a word in front of equilibrium here, though, and that is dynamic, okay? So through the fluid mosaic model worksheet, hopefully you saw that dynamic means changing. Okay, this is a great vocabulary word in general. It's a very common word to use when you're describing something as changing. It's a big ACT word. Okay, it appears on the ACT quite a bit. So diffusion is going to occur until dynamic equilibrium is reached. What does that mean? Well, what that means is that this solution is still never going to be completely still. Remember, particles are always in motion. Okay, so even if we look at a glass of water, let's say, with a red dye in it, and it appears still, it's not completely still. Okay, so those particles that we see there of the red dye and of water are constantly moving around. Okay, so then, by definition, equilibrium, this is the point at which the dissolved particles fill a space uniformly and the concentration of the substance is uniform throughout. But again, it's going to be dynamic and that solution is going to constantly be changing, okay? So this is one example of diffusion. Another good example is that if I were to stand in the front corner of my classroom, and spray perfume, okay? Then you have to imagine that whoever's closest to me is going to smell that perfume first. Whoever's furthest from me, okay, completely across the room, they're going to be the last ones to smell that particular scent because it takes a little while for the molecules to diffuse throughout the room, okay? Diffusion does not have to involve a membrane, but it can. Okay, and that's important to understand. I've given you the example of this happening in just a beaker of water, in the room with a perfume, for example, or some sort of scent, but it can involve a membrane. Okay, that being said, we're going to get to the next type of movement has to involve a membrane. All right, so I just said that osmosis, this is our second type of passive transport, so we're still under that passive category. And this has to involve a membrane, right? So this is where we're going to talk a lot about solutions, concentrations, the plasma membrane in a way that is sometimes kind of difficult to understand, okay? So again, oh, and I forgot to say what net movement meant. So again, net movement means we're talking about overall which direction are particles going to move. Particles can cross the membrane in the opposite direction from what they typically would do, but that's going to be very few, okay? So I'll kind of explain that again eventually. This, in general, though, is the more difficult of these three different types of passive transport for most students. So you might want to just jot some extra things down that I say here, even if I don't write them down. So this is the net movement of water. Notice it says water. So this is only water moving across a semi-permeable membrane, has to involve a membrane, from where water is more concentrated to where water is less concentrated, which sounds really weird, okay? So we're more concerned with where is there more free water versus less free water, right? So that being said, even though the tails of the plasma membrane are nonpolar, water is really tiny, and so it can actually go through the nonpolar regions of the membrane, even though it's a polar substance, okay, because it's tiny. So this is going to happen when big polar substances cannot cross the membrane and the cell needs to get to equilibrium, okay, that the cell is not or the homeostasis, or sorry, I'm getting a little tongue-tied. The level of homeostasis in the cell is not being maintained. Osmosis will occur in order to get the cell to equilibrium if there's a large polar substance on one side of the molecule, or sorry, one side of the membrane that cannot cross. All right, so in general, okay, if we look at this picture, notice that we have a high concentration of water on the top side of this picture, lots of free water molecules in blue, okay? On the bottom of this picture, we have a very low water concentration. We have a few blue waters, we have a lot of sugar down here, okay? We only have 2 sugar molecules up here. This could be sugar, this could be salt, it can basically be any sort of type of ion. We tend to talk the most about sugar and salt, just kind of generally, to cover the topic of osmosis, okay? So when polar solutes are mixed with water, the universal solvent, they're going to form weak bonds with those water molecules around them, okay? So it says, therefore, those water molecules are no longer free to move across the membrane, right? Because essentially what you have to imagine, and let me just circle this one here, if this sugar binds to that water, suddenly that water is huge. It's stuck to the sugar and it can't get across the membrane. And so let's say neither could this one, right? So that being said, okay, anytime you have a solute dissolved in water, every single water molecule that gets bound to that solute, okay, is no longer free to cross, okay? And so I'm just circling a couple of these as examples. So imagine that happening with the rest of those sugar molecules. So then there's way more free water up here, right? So there's lots of free water up here, and as a result of that, notice your arrow here and here from those 2 waters, then water is going to cross to the opposite side of the membrane in order to get this cell to a situation of dynamic equilibrium. Okay, now that being said, some waters could randomly slip across the membrane in the opposite direction, because that's just what's going to happen. Particles are always in motion, so there may be some that go across, but our net or our bulk or overall flow is going to be from the top of this picture to the bottom of this picture in order to equalize these 2 concentrations on either side of the membrane, right? And again, it's these nonpolar tails that are going to separate the cell from its surroundings, okay? And then remember, that's the head of the phospholipid. You just don't see any protein shown here, okay? So there's more free water up here than there is down here. So water is going to flow down its concentration gradient, right? And so that being said, water is going to move toward the higher solute concentration as it's moving down its concentration gradient. So let me go back to that picture, okay? Here's the more concentrated side of this membrane. There's way more sugar dissolved in the water over here than there is over here. So water is going to move down its own concentration gradient as it's moving toward the higher solute concentration. And in this situation, moving toward where there's more sugar, okay? So as a result of that, as water flows down in that picture at the top, extra water is going to accumulate on what's going to be called the hypertonic side of the membrane, and that's going to exert a great deal of pressure on that side of the membrane, and it happens to be called osmotic pressure. So let me go back to this again, okay? So as the water comes from the top to the bottom, okay, then extra water molecules are going to build up over here, and that's going to put pressure on this bottom side of the membrane. And so we're going to talk about what's going to happen as a result of that. Okay, so in general, cells can be in different types of solutions, right? So they can be in solutions that are isotonic, hypotonic, or hypertonic to the inside of the cell. Okay, iso means the same. Okay, hypo means below, and hyper means above. Okay, like think hyperactive. If somebody has hypothermia, their body temperature has dropped below what is normal, might be dangerous. Okay, if they're, I don't know, in a car wreck in the middle of a snowstorm, okay, then their body temperature could drop really low if they get stranded and nobody can find them, just as an example. Okay, so we're going to go through what happens to a cell in each of these three situations. All right, so if a cell is in a situation where it is in an environment that's isotonic to the inside of the cell, then this is the just kind of generalized picture we're going to start with about how to determine what's going to happen as a result of that. So inside the circle is the cell, and outside the circle is the environment. In this case, water is going to be these green squares, and salt is going to be these pink triangles. Okay, you are not really counting the same way that I'm going to explain about you counting every single time you do this. You've got another sheet that we're going to kind of talk through a little bit as well. Okay, but it says the concentration of the solutions inside the cell and outside the cell are the same, right? So as a result, let's see how many waters we've got inside. 1, 2, 3, 4, 5, 6, 7, 8 waters on the inside. There are also 8 waters on the outside, right? There are 6 salts on the inside, and there are 6 salts on the outside. So the concentration on either side of the membrane is the same, okay? So there's not going to be any net movement of water into or out of the cell. However, water can actually flow in both directions, right? So you might have a water that goes out, maybe another one that goes out, maybe then you have a couple that go in, okay? It's just going to be that randomly particles are going to move in and out of the membrane in this situation, or I should say waters are going to move in and out, okay? But there's no big influx of water in, nor does a lot of water leave the cell. So the size of the cell is going to stay the same, and the pressure inside the cell is going to stay the same. This cell is already, okay, in a situation where it's at dynamic equilibrium, right? I think I spelled that correctly. Yep, okay. All right, so then what happens if a cell is put in what's called a hypertonic solution? So here, if you count again, we've got 8 waters on either side of the membrane. In this case, we've got 7 salts on the outside and 4 salts on the inside, okay? So if you imagine binding a salt with a water, right, then there's going to end up being more free water on the inside of the cell than there is on the outside of the cell, okay? And so as a result of that, what's going to happen? Well, water's going to leave the cell, okay? The cell, oops, yeah, you can see that, sorry. Water leaves the cell, okay? The cell is going to get smaller, and pressure inside the cell decreases. Okay, and I would suggest that you write all that on your own notes, okay? So again, this is a higher concentration of solute on the outside of the cell than on the inside of the cell. So there's more free water on the inside. Water is going to move down its concentration gradient and leave the cell. The cell is going to get smaller. The pressure inside the cell is going to decrease, okay? So this outside environment is hypertonic to the inside of the cell. And then in this last situation, if the cell is in a hypotonic solution, where the concentration of the solution outside the cell is lower than that inside of the cell, we're going to determine what's going to happen here. So in this case, we've got, it looks like 7 waters on either side of the membrane. We've got 4 salts on the outside, and we have 6 salts on the inside. Okay, so again, if we imagine every salt that's present bound to a water, that's going to mean that there's going to be more free water on the outside than on the inside. Okay, so what's going to happen? Water is going to flow into the cell. Okay, water is going to move in. Okay, that means that the cell is going to increase in size, and that the pressure inside the cell increases. Okay, so water is moving down its concentration gradient from where there's more free water on the outside to where there's less free water on the inside. So it's moving toward the hypertonic side of the membrane and away from the hypotonic side in an effort to equalize the concentration on either side of the membrane. And that'll happen until the cell gets to dynamic equilibrium, we hope. Okay. The last type of passive transport that's going to occur is called facilitated diffusion, right? The word to facilitate means to help in some capacity or to assist. So this is a type of diffusion through a specific through a specific membrane protein channel. And it's going to be that each type of protein channel that assists a molecule moving by this method through facilitated diffusion, it's going to move one sort of molecule through one specific protein. Okay, so in this case, this specific protein channel is designed to move glucose. It's going to move glucose from high concentration to low concentration. So glucose is a huge molecule, right? But at times, glucose does need to cross the membrane of cells. For example, when you eat, okay, and then you metabolize, say, starch from a piece of toast, from some cereal, some potatoes, something like that, and it gets broken into glucose, then that glucose in your blood needs to get into your cells. And so it's going to cross a membrane protein in order to get from the bloodstream into cells in various organs of your body in order to be metabolized and broken down in those cells and generate ATP. Okay, so sometimes big molecules can cross the membrane. If they're doing it, they have to go through some sort of protein, and in this case, facilitated diffusion is going to be one specific molecule with one specific type of protein, still moving the molecule down its concentration gradient, because this is passive transport through a protein from high concentration to low concentration. All right, so then we keep active transport really, really simple in this course. So we said passive transport is going to move the substance, whatever it happens to be, from high concentration to low concentration. And there's several types of passive transport: diffusion, osmosis, and facilitated diffusion. For active transport, the substance is going to be moved against the concentration gradient, and that is not what molecules typically do. Therefore, the cell is going to need to use energy in the form of ATP. Okay, so if we look at this picture, you've got a molecule that needs to cross the membrane. It's kind of shown on either side. This is like the before up here, and then this is the after down here. Okay, so it's in low concentration on top of the protein. It's in high concentration beneath it. Okay, but it needs to move from low to high, right? So as a result of that, you picture that one of these little blue circles, whatever this happens to be, is going to come into this protein. That's going to cause the cell to use some energy, and then the protein is going to change shape, and it's going to kick that molecule out on the opposite side so that it keeps this molecule in high concentration on the opposite side of the membrane. Okay. Now, with active transport, if the molecule is small, then it's often compared to some sort of pump. Okay, the sodium-potassium pump in our bodies is a good example of this. Basically, sodium and potassium need to be in high concentration on different sides of a membrane, and so our cells expend a ton of energy in order for that to occur. And this is going to play a role in how our nervous system functions. In order to allow our neurons to fire, these concentrations have to be maintained by these sodium-potassium pumps. So we use a lot of ATP for that, okay? I've got a video clip that hopefully we will watch about that. Then, if we're talking about active transport of large molecules, there's two categories. There's endocytosis and exocytosis. Endocytosis is the process of taking some sort of material into the cell by little pockets or enfoldings of the cell membrane. Exocytosis is the process by which a cell gets rid of large amounts of material, like, some sort of waste, through an out pocket or an outfolding in the cell membrane. Within the category of endocytosis, there are two types of endocytosis, phagocytosis and pinocytosis. So pinocytosis, this is a process where cells are going to take in liquid from the surrounding environment into a little end pocket or enfolding. Phagocytosis is a process where extensions of cytoplasm are going to surround and engulf a large particle and take them into the cell. And then an example of that is an amoeba surrounding and engulfing its food, white blood cells in your body, phagocytize things that are pathogenic, like bacteria and viruses, for example, that might cause some sort of infection. Okay, and so we'll watch a couple of videos about these as well. The definitions are just pretty straightforward. It helps to watch videos instead of me trying to explain things that you kind of need to wrap your brain around visually. Let me know if you have any questions.